My name is Claudia and I'm a student at the University of Kent studying biology. I applied for the 10-week studentship because I wanted to explore whether I would want to specialise in plant pathology at a postgraduate level. My project focused on the relationship between biofilm formation, bacteriophage biocontrol, and pathogenicity of Pseudomonas syringae. This pathogen causes bacterial canker disease on cherry and is the biggest issue affecting commercial growing of cherries. In the summer, bacteria live as leaf epiphyte, possibly in a biofilm form. In the autumn, bacteria washed from the leaves infect leaf scar wounds, which leads to canker lesions and dieback that can kill the trees. I began growing biofilms using 25 different strains of Pseudomonas syringae in both glass tubes and in plastic 96 well plates. I compared glass tubes and different brands of the well plates in terms of throughput, consistency, and speed of biofilm formation. The plates of the best biofilm growing parameters were used to compare biofilm forming capacity of the selected Pseudomonas strains at different time points and at different bacterial concentration. In addition, I've also quantified the attachment capability of the selected strains at different time points and in different conditions. I compared the biofilm formation of selected strains on the cherry leaves under different relative humidity conditions to find the best and worst biofilm forming strains. This information is crucial to know before we can test how effective the phages are at destroying biofilms. We can now track at which time point the biofilm forms for each strain. Once we know which phages control biofilm formation of which Pseudomonas strain and to what extent, then we can choose the most appropriate phages to control Pseudomonas syringae on the leaves in the summer and reduce autumn infections in the commercial growth environment. I have learnt how experiments are planned and designed, how bacterial cultures are prepared, and the importance of testing strains against each other only once we have ensured these are at the same concentration, by using the spectrophotometer and plating serial dilutions to calculate colony-forming units. I learned aseptic techniques as contamination was all too easy to get, so I tweaked my method over the weeks to be more and more careful. I learned how to use different machines and technologies that I previously had no experience of using, such as the microplate reader, autoclave, and fluorescent microscope, which I found particularly enjoyable to use and capture images of the GFP-tagged biofilms. I'll be undertaking a year in computing before my final year in biology, in which I now feel inspired to choose a bioinformatics project on a biocontrol topic for the main coursework. I would like to thank BSPP for giving me this opportunity and my supervisor, Dr. Matevz Papp-Rupar, for mentoring me.